All right, class. So we are in chapter 2 still. Part A was a little bit about the basics of chemistry, and feel free again to watch that recorded lecture on the YouTube channel. This will be part B of chapter 2. And again, a lot of these chapters are divided into several parts. They can get kind of lengthy, but we'll get through all of this part B and then be good for this lecture in this chapter. So we'll talk a little bit about biochemistry here. So we talked about chemistry, and now we'll relate it to biology, which is the study of living things. But if we combine biology and chemistry together, we get biochemistry, which is the study of chemical composition and reactions of living matter. And it's really important to study biochemistry or have a little background of it going into physiology, because we'll be really kind of studying in depth the physiological reactions of many processes in the body. So we'll talk about all chemicals being either organic or inorganic. And an inorganic compound does not contain any carbon. So the inorganic compounds will be water, salts, many acids and bases. If a compound is organic, it will contain carbon. They're also much larger and all covalently bonded, meaning they're sharing electrons. So organic compounds are things like carbohydrates, fats, proteins, nucleic acids. Just larger compounds are generally organic, and they can be broken down into their specific parts. Both of these are equally essential for life. We'll talk a bit about water, because water is very important for a lot of different reasons. It's the most abundant inorganic compound, and it accounts for about 60 to 80% of the volume of all living cells. So all of your cells are mostly made of water. It has other important properties like a high heat capacity, a high heat evaporization, polar solvent properties, meaning water has the ability to surround substances in order to dissolve them very easily because of its polarity. It's very reactive, and it provides a cushioning kind of protection throughout the body, whether that's around your brain or organs. The high heat capacity of water means it has the ability to absorb and release heat with little temperature change. So water is a really good, when we think about homeostasis, it provides a good balance to prevent sudden changes in temperature, because water needs to get extremely hot to boil and extremely cold to freeze. So it has this ability to prevent change in temperature. It has a high heat of vaporization. Evaporization requires large amounts of heat, and it's also a useful cooling mechanism. And we know that through sweating. Sweating cools off the body, and that's due to the properties of water. The polar solvent properties, like we talked a little bit about, has the ability to dissolve and dissociate ionic substances. So that means substances that have charges to them. Because water is a polar molecule, it has charges to it. It's able to kind of dissociate or break apart ionic substances like salt, sugar. It forms hydration or water layers around large charged molecules like proteins. And water is your body's major transport medium. So here's a look how water dissociates around salt. So here's the polar water molecule. If you watched the previous lecture, I talked about polarity of water. That just means one end of water is more negative by the oxygen, and the other end of water is more positive by the hydrogens. And what this polarity allows it to do, it's kind of shaped like a V, and it has the ability to surround, its negative end surrounds positively charged sodium, and the positive hydrogens will surround the chlorine negative ions. So it has the ability, because it's polar, to break apart these ionic compounds because the opposites attract. So the opposite charges of water will attract the opposite elements of this compound and break it apart really easily. And that's how we can dissolve eventually salt in water. It's reactive, so it's a necessary part of hydrolysis and dehydration synthesis reactions. And that just means that water is either added or subtracted to these reactions. Cushioning, it provides certain organs protection from physical trauma. Like your cerebral spinal fluid will cushion your nervous system organs, the brain, the spinal cord. A salt then is an ionic compound. And again, ionic just means it has two charges to it. It will dissociate into its separate ions in water, and it will separate into cations, which are the positively charged molecules, and the anions, which are the negatively charged. This doesn't include hydrogen and hydroxide ions. Those are separate. Salts, all ions of salts are called electrolytes because they can conduct an electrical currents in the solution. Ions play a very specialized role in body functions. Sodium, potassium, calcium, and iron, those are the big ones that we'll talk about in this class. The sodium potassium pump. Calcium is important in muscle contraction, and iron is important. It helps make up hemoglobin, which transports your oxygen. This ionic balance is very vital for homeostasis. So keeping these ions balanced in the body is incredibly important, as we'll find out. Here are some common salts in the body: calcium carbonate, sodium chloride, potassium chloride, and calcium phosphates. Acids and bases are both electrolytes, so that means they will ionize or have a charge and dissociate in water. We often call acids a proton donor because they will release their hydrogen ions. So acids will have a lot of hydrogen ions to them, and we often call a hydrogen ion a proton donor. So that's why we say acids are proton donors. They release hydrogen ions. An example is hydrochloric acid, and it will release its hydrogen and a chloride ion. Some of these important acids are hydrochloric acid, acetic acid, and carbonic acid. A carbonic acid will play a role in how carbon dioxide is transported in the blood. So we'll come back to that one later on in this semester. And then a base, we call them proton acceptors. So they will pick up excess hydrogen ions in a solution. When a base dissolves in a solution, it will release a hydroxyl ion, which is abbreviated OH negative. So if you can remember, a base accepts protons, they'll pick up excess hydrogen ions in a solution. And some important bases are bicarbonate ion and ammonia. So this brings us to the pH scale. If you're wondering where we're going with all this, the pH scale just gives the acid-base concentration of a solution. So the pH scale specifically measures the concentration of hydrogen ions, which again are these H+, the concentration of hydrogen ions in a solution. And the more hydrogen ions that are in a solution, the more acidic that solution is. pH is what we call a negative logarithm of hydrogen ions in moles per liter of concentration. That ranges from 0 to 14. It's a logarithmic scale, so each pH unit represents a 10-fold difference. What that just means is that, for example, if you have a pH of 5 solution, that means it's 10 times more acidic than a pH 6 solution. So everything has a 10 times difference in terms of concentration. Acidic solutions, again, have very extremely high hydrogen ion concentrations, but they have a low pH. So there, if a solution is acidic, its range will be 0 to 6.99. A neutral solution just has an equal number of hydrogen and hydroxyl ions. So that neutral solution will always be a pH of 7. Pure water has that pH neutral solution. So sometimes that's become a marketing ploy where people will market their bottles of water, say neutral pH levels, to try to make you to want to drink it. And it probably is better to drink completely neutral water. So you could spend extra money and buy that water. pH of pure water then, again, has a 7. Then an alkaline or a basic solution has low hydrogen concentrations, but a high pH. So an alkaline or basic pH range would be anything over 7.01 all the way up to 14. And this is the pH scale and pH values of different representative substances that you might find in your house. Or you might not. We have sodium hydroxide is incredibly basic. So basic is at the top where it says 14, and And then we work our way down. Neutral, again, is right around blood and milk. So water would be at neutral. Blood is just a little bit basic at 7.4. And milk is just slightly under 7, so slightly acidic. You have egg whites, household bleach, household ammonia, oven cleaner. So you can kind of see where just basic household items fall along this scale. Hydrochloric acid is extremely acidic. You do not want to spill that on your fingers. That'll create a sore in your skin. Lemon juice is very acidic. Wine is also acidic. Black coffee can be acidic too. So a neutralization reaction is when we mix acids and bases together and a displacement reaction occurs, forming a water and a salt. So this is just a special type of chemical reaction that whenever you mix an acid and a base, you can kind of see here what happens. We have a displacement reaction. So again, these are the reactants on the left side of the equation, products on the right, and the letters or abbreviations of these elements just flip-flop. So the chloride goes where the OH is, and the OH goes where the chloride is. So what we form is sodium chloride, which is a salt, and water. So this is just a neutralization reaction. We can mix a really strong acid and a really strong base together. On their own, they'd probably be really dangerous to touch and handle, but when we mix them together, we can neutralize them to form salt and a water. A buffer is very important because buffers will resist abrupt and large changes in pH. So you have buffers in your body to help resist pH changes. Buffers can release hydrogen ions if pH rises or becomes more basic, and it can bind hydrogen ions if the pH falls or becomes more acidic. So it can convert really strong acids or bases into weak ones. An important buffer system in your blood is the carbonic acid and bicarbonate system. I don't know why these letters are here. That should just be like an arrow. But what we have is a weak acid, and it can become a weak base and a proton donor through this carbonic acid and bicarbonate system. What this buffer system is important for is carrying carbon dioxide in the blood, and it will change into either a weak acid or a weak base in order to carry the carbon dioxide through the blood. When we get to respiratory system, we'll talk more about this system, but I just want you to know that buffers are important in your body because they help resist changes in pH that could be occurring, whether that's through hormones or some sort of other imbalance that's going on. So we talk a little bit about a clinical significance here. We'll talk about enzymes. An enzyme in the body will work within a very narrow pH range. So again, in the first part of this chapter, part A, we talked about how enzymes are necessary just to increase the rate of a chemical reaction. They're never used up. They never change the product of the reaction, but enzymes are just really important in the body to help to get chemical reactions going a little more quickly. And different enzymes are viable or work within extremely narrow pH ranges. So for example, if you have an arterial pH of 7.0 during cardiopulmonary resuscitation, this will predict a poor outcome because the enzyme isn't able to work during that. Patients presenting with an arterial pH of less than 6.85 will rarely survive. So if you're having some sort of heart failure and you have a difference in pH, but if the pH is just narrowly lower, that enzyme to help that part of the body function will not work. So that's just what this imbalance is talking about, just extremely, really slight changes in pH in the body can cause really severe outcomes, and that's why enzymes work within a very narrow pH range. We'll talk a little bit now about organic molecules and hydrolysis. Organic molecules all contain carbon. The exceptions to that are carbon dioxide and carbon monoxide, which are inorganic. Carbon is what we call electron neutral. It completely shares all of its electrons. It never gains or loses them. And that's because of its spot in the periodic table, it has the ability to form 4 covalent bonds with other elements. Carbon is really unique to living systems. Carbon is probably one of the most important elements in the body. It helps form major organic compounds of carbohydrates, lipids, proteins, and nucleic acids. And we're going to talk a little bit about those organic compounds. Many of these are polymers, and a polymer, poly just means many. So a polymer means many of these organic compounds are chains of similar, smaller units called a monomer, which is a singular one building block. Polymers are synthesized. That means they are created by dehydration synthesis reactions. A dehydration synthesis reaction means they take out a water, they dehydrate the reaction to create it. And polymers are broken down or broken apart by hydrolysis reactions. And that just means a water is added in to break it apart. So this just takes a look at a dehydration synthesis reaction. What it's doing is it's taking 2 monomers, and again, a monomer is any single element. And if we take water out of it, as you can see here, if we take out an H2O, which is shown in blue here, it allows the 2 monomers to be linked by a covalent bond with just that oxygen there. So a dehydration synthesis basically just takes out extra elements and atoms that you don't need in order to bind 2 monomers together. A hydrolysis reaction just does the opposite. So we have monomers linked together by a covalent bond shown here. And if we add in a water, you're adding in 2 hydrogens and 2 oxygen and an oxygen. And those 3 elements have to go somewhere. So what they do is they break apart the bond that's holding the 2 monomers together, and it completely breaks that apart. And this is just an example reaction combining glucose and fructose, which are 2 monomer sugars. And when we add them together, we create a polymer, a sugar, or also called a disaccharide, meaning 2 sugars have been linked together. And the special name when you combine a glucose and a fructose together is called sucrose. We're getting into a lot of chemistry. I was a chemistry major. Some of you might know that. So this interests me, but I don't expect you guys to know all those details. Carbohydrates include sugars and starches. They contain carbon atoms, hydrogen and oxygen atoms, and about that ratio. There's 3 classes of carbohydrates. A monosaccharide means 1 sugar. A disaccharide means 2 sugars. And poly, again, means many sugars. So a polysaccharide is just a polymer that's made up of monomers or 1 units of monosaccharides, so just single sugars. Monosaccharides are simple sugars. They contain 3 to 7 carbon atoms, usually in some sort of hexagon or octagonal shape. Here's the general formula, with N being the number of carbon atoms. Monosaccharides are just monomers of carbohydrates. Some important monosaccharides are pentose or hectose sugars. And pentose looks like a pentagon shape, and a hexose sugar has kind of a hexagon shape. The pentose sugars are ribose and deoxyribose, which will make up your DNA and RNA. And a hexose sugar is glucose, which is the main important blood sugar in the body. So these are just examples of a monomer of a carbohydrate. And again, a monomer just means 1 single one. And you can see here, the hexose sugars have more of the hexagon or 6-sided shape, and the pentose sugars have the 5-sided or pentagon shape. So chemistry is a lot about shapes too. A disaccharide is just 2 double sugars. They're too large to pass through a cell membrane. Important disaccharides are sucrose, maltose, and lactose. And it's formed by the synthesis of 2 simple sugars together. So that's a disaccharide. And here's some common important disaccharides in the body: sucrose, maltose, and lactose. And again, a disaccharide just means 2 sugars bounded together. And then a polysaccharide just means many sugars bound together. Important polysaccharides are starch, which is the carbohydrate storage form used by plants, and glycogen, which is the carbohydrate storage form used by animals. Glycogen is just the way that your body stores glucose, and it stores glycogen in the liver. And they are not very soluble, meaning they don't dissolve easily. So here are carbohydrates and their molecules and how they're important in the body. Basically, this is a long molecule of glycogen, which is just the polymer or the polysaccharide of all these simple single monosaccharides or simple sugars bound together. So this is a polysaccharide. It's just a long chain of linked monosaccharides or simple sugars bound together. And then a lipid will contain carbon, hydrogen, and oxygen, but much less than in carbohydrates, and sometimes it'll contain a phosphorus. Lipids are insoluble in water, so another word for a lipid is a fat or an oil. These are the main types of lipids: triglycerides, phospholipids, steroids, and eicosanoids. Triglycerides are called fats when they're solid and oils when they're liquid. A triglyceride, that word tri means it is made up of 3 fatty acids bonded to one glycerol molecule. And glycerol has the word, has the prefix that has to do with sugar in it. And it ends in an "-al", like alcohol. So it's like a sugar alcohol. And the main functions of your triglycerides are to help store extra energy that your body needs, insulate the body, and protect the body. So this is just a look at a triglyceride. What this is showing, these are the, this is the fatty acid, it will contain 3 fatty acid tails, as you can see here. There's the triglyceride. I'm sorry, my slides take us one at a time. Here's the triglyceride, and these are the 3 fatty acids on the bottom in yellow, and the glycerol compound is at the top. And here's an animation about fats. Again, you can download this from Canvas and watch that animation on your own. Triglycerides can be constructed of saturated fat, which means all the carbons are linked via single bonds. And these will create a linear molecule, which can pack closely together, which form a solid at room temperature, like animal fats or butter. So this is what a saturated fat looks like. What it refers to is all of this line here, this kind of diagonal line. Each point means there's a carbon atom at that point. And because they're completely single bonded, it means it's a completely linear kind of structure. So all of these fatty acid tails can stack on top of each other really nicely. They fit together kind of like in an accordion. So they can create butter at room temperature or a solid. This is also why saturated fat is not good to eat, because you're eating kind of this compacted fat that's a lot harder for your body to break down because they can be compacted really easily on top of each other. Versus unsaturated fat. Unsaturated fatty acids means that the carbons are linked via double bonds, and the double bonds cause a kink in the fatty acid tail, so they just can't pack together as closely. So most unsaturated fats are liquid at room temperature, like olive oil. A trans fat is a modified unsaturated fat that resembles a structure of saturated fat and is considered healthy. And if you hear of omega-3 fatty acids, those are considered more heart healthy. So this is a look at an unsaturated fat. This double bond just allows the fatty acid tail to kind of kink or kind of go downward, meaning the fatty acid tails cannot pack together as tightly, which is a good thing. Your body is able to break those apart a little more easily. A phospholipid is just a modified triglyceride. It has a head and a tail region, which have different properties. If you remember learning about the plasma membrane on your cell, it's made up of phospholipids. The head is always polar, and hydrophilic means it's attracted to water or loves water. And the tails of phospholipids are nonpolar and hydrophobic, meaning they're repelled or they dislike water. And they are, like we said, really important in cell membrane structure. So this just shows the phospholipid structure with the polar head, which is this phosphate group, and the nonpolar tail. And nonpolar means water-fearing or hydrophobic, and the polar head means water-loving or hydrophilic. So this shows the polar head and the nonpolar tail, and how the phospholipids line up to create this lipid bilayer in the cell membrane structure. So the tails touch each other, and then the heads are seen on the exterior side. Lipids, so we're gonna keep going talking about steroids. Steroids consist of 4 interlocking ring structures. What I want you to remember about steroid is that the most important steroid in the body is cholesterol. It's made by your liver. It's also found in animal products like cheese, eggs, and meat. Steroids are the starting material for synthesis of vitamin D, other steroid hormones, and bile salts. So that's why steroids are important, and they're also important in cell membrane structure. So this is what the steroid structure looks like. There are these 4 interlocking rings, and cholesterol is shown here. And the cholesterol is the most important steroid. Eicosanoids are derived from a fatty acid found in cell membranes. The most important eicosanoid are prostaglandins. And prostaglandins are really important because they will help in blood clotting, the control of blood pressure, inflammation, and labor contractions. Inflammatory actions are blocked by NSAIDs. You've probably heard of NSAIDs before. And NSAIDs stand for non-steroidal anti-inflammatory drugs such as aspirin or ibuprofen. Proteins then comprise about 20 to 30% of your cell mass. They are the most varied functions of any molecule. So your proteins have the most varied functions in structural, chemical. They have enzymes, contraction in your muscles. They are polymers of amino acids that are held together by peptide bonds, and their shape and function are due to 4 different structural levels. So structural proteins help in mechanical support. So an example of a structural protein is collagen, which is the single most abundant protein in the body. So collagen is important to give strength to your bones, tendons, and ligaments. You can have enzyme proteins, which are just catalysts. So they're essential for virtually every biochemical reaction in the body. And then you have transport proteins, which are important for moving substances. So this is showing hemoglobin, which helps to transport oxygen. So proteins are incredibly diverse in their structure and their function. You have contractile proteins, myosin and actin, which slide past each other to contract your muscles. Communication proteins transmit signals between cells. They can act as hormones, chemical messengers, or as receptors. And then you have defensive proteins, which are antibodies. So again, proteins are incredibly diverse. There will be a whole chapter on how your body creates proteins, doing DNA, RNA, transcription, translation. And just remember that it's so important to make all those proteins because they're so diverse and they're important for every aspect in your body. So proteins are made of amino acids, and you have 20 types of amino acids in your body. And proteins are just made by different rearrangements and combinations of those 20 types of amino acids. These amino acids are joined by what we call peptide bonds that contain an amine group and an acid group, so they can act as an acid or a base, and they differ by which of 20 different R groups is present. So what that just means is that each amino acid takes on this structure. And again, you have 20 amino acids in your body. And the only difference between the 20 amino acids is what we call the R group. And that is shown, this R in green is pointing at this orange circle. So this is the R group, and that's the only difference between the amino acids. So all proteins are made up of these amino acids. They're linked together by peptide bonds. You don't really need to know how that happens. Again, it uses a dehydration or hydrolysis reaction. But basically what a protein does is it takes different amino acids and binds them together, and it produces this peptide bond between them. The structural levels of protein, so you have 4 levels of protein structure which determine its shape and function. The primary structure just means the linear sequence of amino acids. So that just means the order of amino acids that are being placed together with peptide bonds. The secondary structure describes how these primary amino acids are interacting with each other. So this kind of line of amino acids can coil together to create an alpha helix, or they can kind of be pleated sheets that look like an accordion ribbon, and those are called beta sheets. So that's the secondary structure. The tertiary structure describes how the secondary structures interact with each other. And then the quaternary structure describes how two or more different polypeptide chains, so many amino acid chains, interact with each other. So I'm sure you're all a little confused by now. Here's the primary structure, which just means a line of amino acids. So you have amino acid here, and again, the only difference between each amino acid is shown in this green R group, which will have its own structure. So primary structure is just the line of amino acids. The secondary structure, the line of amino acids will then either coil on itself to create an alpha helix, or kind of fold on itself to create a beta sheet, which is like a pleated sheet that's just folds on itself. So that's the secondary structure. The tertiary structure just shows the secondary structures interacting, whether beta sheets or alpha helixes are bound together, sometimes in a compact globular molecule. And then the quaternary structure just kind of throws in two or more polypeptide chains at the same time. So that gives the protein its different structures, and you can watch a great animation on that to help you with that to understand it a little bit better. Shapes of proteins can be either fibrous or globular. Fibrous are structural proteins. They're more strand-like, water insoluble, so they do not dissolve in water. Most of them will have a 3D structure to them, and they provide mechanical support and tensile strength. And an example of these are keratin, elastin, collagen, and certain contractile fibers. Globular or functional proteins are compact, spherical. They can dissolve in water. They're very sensitive to environmental changes. They will also have this 3D shape, and they'll have specific functional regions, which we call active sites. So your globular proteins will be anything that can bind to something else, so like an antibody, a hormone, any other molecular chaperone, we call them, or an enzyme. Proteins can be denatured, which means they can just be deactivated at any time. And denaturation means that globular proteins will unfold. And when they unfold, if they lose their shape, that means they'll become deactivated. The fibrous group of proteins are a little bit more stable, but globular proteins can unfold. And when they unfold, they'll lose their shape, they'll become deactivated, and their active sites will be no longer active. This can be caused by acidity. So if you have a decrease in pH or an acidic environment, that can denature or deactivate a protein. It can also be caused by increase in temperature. So extremely hot temperatures can denature proteins. It's usually reversible, so the protein can go back to being active if normal conditions are restored, but it is irreversible if changes are very extreme. And an example of that is you can't undo cooking an egg, you can't put an egg back in the shell. An enzyme is a globular protein that acts as a biological catalyst. Catalysts, like we talked a little bit about with enzymes, they regulate and increase the speed of chemical reactions without getting used up in the process. And what they do is they just lower the energy needed to initiate a chemical reaction. So if you think of you're trying to get over if you're trying to travel up a large hill with your car, a catalyst or an enzyme, what it does is it just lowers the height of that hill so your car can get over the bump more quickly. Enzymes, most functional enzymes consist of two parts, the apoenzyme and the cofactor. Enzymes are very specific. That just means they only act on a very specific substrate, so they can only bind to a certain substance. And most enzymes, they usually end in ASE as their suffix, and they're often named for the reaction that they catalyze. So a hydrolase, oxidase, those are all enzymes. The enzyme action, they will lower the activation energy, which is the energy needed to initiate the chemical reaction. So they'll prime the reaction, you could call it. They allow the chemical reaction to proceed quickly at body temperatures. You have 3 steps involved in enzyme action. Basically, these 3 steps just involve the substrate binding, the complex undergoing a rearrangement, and then a product being released from the enzyme. So this just shows, I gave the example of getting your car up a large hill. This is kind of a visual representation of what an enzyme does to lower the activation energy. So if in order to a chemical reaction to occur, the reactants have to get over this energy of this hill. And what an enzyme does is it just lowers the energy requirement needed for those reactants to become products. Mechanism of an enzyme is again described here in these 3 steps. So if you're watching the recording, feel free to pause the recording to take a look at those 3 steps again. And these slides take you through one at a time. Nucleic acids then are the largest molecules in the body. They're composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus elements. They are nucleic acid polymers are made up of monomers called nucleotides. And the 2 major classes of nucleic acids are DNA and RNA that we'll get into a lot more depth when we talk about how they're made. But just know that DNA and RNA are these 2 groups, 2 classes of nucleic acids. DNA is important because it holds your genetic blueprint for the synthesis of all proteins. DNA looks like a double-stranded helical molecule located in your cell nucleus. And the nucleotides will always contain a deoxyribose sugar, a phosphate group, and one of these 4 bases, adenine, guanine, cytosine, or thymine. So that is DNA. The DNA, the bonding of the nitrogen base from the strand is opposite. So what that just means is that those 4 nitrogenous bases only pair 2 and 2. So adenine will always only be bounded or pair with thymine, and guanine will always pair with cytosine. And again, we're going to come back to DNA. And if you've taken other cell biology courses, you probably know what I'm getting at here, but we'll come back to DNA. But first, I just want to tell you a little bit more about the structure. You can see the double helical strand, the double helix model shown here. And if you look really carefully, you see these 4 bases, adenine, thymine, cytosine, and guanine, and how they are bounded together complementary. So thymine always is bound to adenine, and guanine is only bound to cytosine, and they're bound with hydrogen bonds. RNA will then link DNA to protein synthesis. So RNA is slightly different. We'll talk about why it's important. It's important because it contains the ribose sugar, and instead of containing that thymine, nitrogenous base group, it has uracil. There's 3 different varieties of RNA that help carry out DNA orders for protein synthesis, and we'll talk more about those at a separate time. This would be a great animation to watch as well, kind of just showing the differences between DNA and RNA. RNA is always single-stranded, as you can see there. ATP then, ATP, so again, if you're wondering where we're going with all this, we're giving you kind of a brief overview of the biochemistry and the language needed of biochemistry to talk about the different elements and compounds when we get to bodily functions and processes. ATP is very important. It's your body's currency system for energy. Chemical energy is released when glucose is broken down and is captured in ATP, which is adenosine triphosphate. ATP always directly powers chemical reactions in the cells. It will offer immediate usable energy needed by your body cells. Without ATP, your cells die and you die. So mitochondria create ATP. Oxygen is required for making ATP. So without oxygen breathing, you die within minutes because your body isn't able to create the ATP or energy it needs to power all of the chemical reactions. ATP we'll talk about here. It has an adenine group, the ribose group, and 3 phosphate groups. And these 3 phosphate groups are bound together with very high energy phosphate bonds. And when those bonds break, that releases energy. So that's where the energy comes from ATP. It comes within these phosphate groups bound together. The terminal phosphate group of ATP can always be transferred to other compounds that can use the energy stored in that phosphate bond. So if we lose one phosphate group, so you can see 3 phosphate groups here. If we lose one, the name of this compound changes its name to adenosine diphosphate. And if we lose 2, the name of the compound becomes adenosine monophosphate for 1 phosphate group. So adenosine monophosphate, that's what these brackets are showing down here. Adenosine diphosphate and adenosine triphosphate includes all 3 phosphate groups. This shows the hydrolysis of ATP, which just means the breaking down of ATP. So ATP again has 3 phosphate groups, and if we break it apart, if we take off one of the phosphate groups, we'll get adenosine diphosphate, meaning 2, and then 1 is separated, plus a lot of energy that's released when we break apart those phosphate bonds. Here are 3 examples of cellular work driven by energy from ATP, transport work, ATP phosphorylates, the transport proteins, mechanical work in contractile proteins in your muscle cells, and chemical work in different chemical reactions. So I'll stop the recording here. Thanks for listening, guys.